The objective of our experiments are: first, to observe the effect of tonicity towards erythrocytes. Second, to observe the effect of sodium chloride concentration towards the percentage of hemolysis of erythrocytes. Third objective is to determine the concentration of sodium chloride that is hypotonic, hypertonic, and isotonic towards erythrocytes. The final objective is to differentiate between tonicity and osmolarity. Four test tubes are labeled as hypertonic, hypotonic, isotonic, and control. 3 test tubes are filled with 5 ml of corresponding concentration of sodium chloride solution and the fourth one with distilled water. 50 microliter of blood is added into each test tube using micropipette. Each test tube is then covered using parafilm, and the mixtures are gently mixed. All the mixture are then left for 20 minutes at room temperature. After 20 minutes, all the tube contents are mixed again, and later centrifuged for 5 minutes at 200 revolutions per minute. Afterwards, all the supernatant are transferred into different clean test tubes using pipettes. The absorbency of supernatants in each test tube is determined using the spectrometer. The spectrometer is set at the wavelength of 540 nanometer, and distilled water is used as blank to zero it. The value of absorbency of supernatants are used to calculate the percentage of hemolysis using this equation. The whole experiment is then repeated using the following sodium chloride concentration solutions. A graph of percentage of hemolysis versus sodium chloride concentration is plotted. The whole experiment is repeated again using the following solution.
For sections A1, we have a table show the percentage of erythrocytes undergo hemolysis in solutions of different tonicity. How we calculate the percentage of hemolysis? The percentage of hemolysis of the supernatants was calculated using the formula shown. Table 2 show the percentage of erythrocytes undergo hemolysis and the increase in percentage of hemolysis in different concentrations of sodium chloride solution. 2 graphs was plotted based on the data in Table 2. The first graph showed the percentage of hemolysis against different concentrations of sodium chloride solution. Graph 2 showed the increase in percentage of hemolysis against concentrations of sodium chloride solution. From the peak of graph, we can determine the point of lysis of erythrocytes. The point of lysis of erythrocytes is 0.45% sodium chloride solution. Section B. Table 3 shows the percentage of hemolysis of erythrocyte in different types of solution.
In Section A1, the absorbency of supernatant in hypertonic solution and isotonic solution are nearly zero.
In hypertonic solution,
The concentration of non-penetrating solutes inside the cell is lower compared to outside the cell. So to reach equilibrium, the water inside the cell moves out from the cell. This will cause the cell to shrink. The solution after the centrifugation is clear. This indicates that there are no loss of hemoglobin occur.
in isotonic solution,
The concentration of non-penetrating solutes inside the cell and outside the cell are the same. So the movement of water into the cell and out from the cell are the same. This will cause no change of volume of the cell. The solution after the centrifugation are clear due to no loss of hemoglobin.
in the hypotonic solution,
The concentration of non-penetrating solutes in the cell is higher compared to outside the cell. To reach equilibrium, water from outside the cell diffuses into the cell. This will cause the cell to swell. When the cell swells, the pore of the plasma membrane will become bigger. This will cause the hemoglobin to diffuse out from the cell. This is why we found that the solution after the centrifugation is red. This is due to loss of hemoglobin from the cell. For the red blood cell in the distilled water, most osmosis occurs here. This is because the cells are in the most hypotonic solution. It acts as the reference for other solutions. In the experiment section A2, a graph is plotted. The percentage of the hemolysis decreases when the concentration of sodium chloride increases. The point in the graph where the flat line starts is the concentration of isotonic. It means that the concentration that hemolysis starts not to occur. There are some errors that may occur during the experiment that cause the third and fifth point of the graph ran from the line of the graph. The error occur may due to the supernatant that do not mix well before the centrifugation. Besides that, we observe that there are some blood clot in the solution. The clumping of blood will reduce the surface area of the red blood cell which are exposed to the solution. This will cause number of cells reacting through osmosis is less. So the result may be different from our expected value.
In this section, we want to determine the tonicity of solution and its effect on erythrocyte. A solution's tonicity is a measure of solution's ability to change the volume of cells by altering their water molecules.
For 300 milliosmol of sucrose,
The solution is isotonic to the erythrocytes because sucrose is non-penetrating solute. The solution and cytosol of erythrocytes are isoosmotic.
For 300 milliosmol of glucose,
The solution is hypotonic to the erythrocytes because glucose is a penetrating solute, while the solution and the cytosol of erythrocytes are isoosmotic.
for 300 milliosmol of sodium chloride,
The solution is isotonic to the erythrocytes because sodium chloride is non-penetrating solute. The solution and the cytosol are isoosmotic.
for 300 milliosmol of urea,
The solution is hypotonic to the erythrocytes because urea is penetrating solute. The solution and the cytosol are isoosmotic. The urea will move in the erythrocyte due to its concentration gradient. As the result, the percentage of hemolysis should be 100%, which the erythrocytes completely hemolysis.
for 150 milliosmol of urea,
The solution is hypotonic to the erythrocytes because urea is a penetrating solute. The solution and the cytosol are isoosmotic. The urea will move in the erythrocytes due to its concentration gradient.
for 600 milliosmol of urea,
The solution is hypotonic to the erythrocytes because urea is a penetrating solute. The solution and the cytosol are hypoosmotic. The urea will move in the erythrocyte due to its concentration gradient.
for 300 milliosmol of urea, in 300 milliosmol of sodium chloride.
The sodium chloride is non-penetrating solute. The solution is isosmotic to the erythrocytes, while the solution and the cytosol of erythrocytes is hyperosmotic. Urea is more tend to move into the erythrocytes due to its concentration gradient, but it would not give a big difference to the size and volume of the cell. Therefore, theoretically, the cell should not have any changes of volume and size.
There are 5 main points in conclusion. First, the percentage of hemolysis of isotonic solutions, hypertonic solutions, and hypotonic solutions are 1%, 1.08%, and 105.74% respectively. Second, the percentage of erythrocytes undergoes hemolysis decrease with increase in concentrations of sodium chloride solutions and remains constant regardless the increase in sodium chloride concentrations after 0.55% sodium chloride solution. For the third point, the point of lysis of erythrocytes is 0.45% sodium chloride solutions. Number 4. Sucrose, glucose, and sodium chloride are non-penetrating solutes that will influence the movement of water across semi-permeable membrane by osmosis, while urea, which is the penetrating solute, has no effect on water movement. Lastly, osmolarity is determined by the total solute concentrations of a solutions, regardless whether the solute is penetrating or non-penetrating, while tonicity is determined by the concentrations of non-penetrating solute in the solutions that measures the ability of the solutions to change the volume of cells.
Now, we're going to talk about what we have learned throughout this experiment. In this experiment, we have learned the effects of tonicity on erythrocyte and how it actually affects the cell volume. Besides that, we have also learned the difference between tonicity and osmolarity. And we have found that the water movement of cells in and out is affected only by tonicity. We have also learned all about osmosis in this experiment. Thus, we come to a conclusion that there are many complex processes in our body, and by this experiment we realize how important it is to maintain a good health so that everything in the body functions well. Thank you.
Won't you be my, won't you be my, won't you be my Valentine? Because I adore you, I'd give anything for you. You make me smile, smile, smile. And when I'm around you, things kinda get hazy. Because I am crazy, crazy for you. I cannot describe it except that I'm in love. Yeah, I'm feeling feelings, feelings for you.